That would be me if this works. Okay, that's all right. And then you see me. And,
You should turn the.
yes, I know. I connected it. Now, I don't think it's talking. What? I don't need anything. Doesn't have nothing to do with it. Oh, yes. Okay. What? Need anything, do you? Need anything? Need anything? I don't think it's, maybe it's the first speaker. It's not even 2 o'clock yet. Oh, yes. Check. That's possible. So because audio.
Want to listen for e-cigarette?
It's live?
Yeah.
What is it, real or no?
Real. Talk about e-cigarettes and
Who's talking?
Jorge Campbell and 2 people from UCLA. Interesting.
Why is it not connected?
Good question. Want to close the door?
But it's not working.
It's working. All right. Heating.
Okay.
Thanks. Hello, everybody.
Hi, Dr. Freeman.
Hi.
Hello.
Hello, hello.
Hi, everybody.
Hello.
Hello.
Very good afternoon.
Okay. So Jorge, you're going to start. Is Jorge on the call?
Who is going to start first?
I think Jorge was going to start. Oh, strange. Jorge, you on? I don't see Jorge on. Okay, Cameron, do you want to go first then?
You want me first?
Yeah, is that okay?
Yeah, I'm okay. Okay.
Okay, welcome to the next SART program. Today's symposium is entitled Cardiovascular Effects of E-Cigarettes. And we're having 4 people today. We have Jorge Espinoza Durat, Kamral Hassan from our group, and we have 2 from the UCLA group, Rajat Gupta and Jocelyn Castellanos. And there'll be also Dr. Jesus Arujo will be introducing them a little bit. But we'll start with Jorge, and then we'll go on. And Jorge is sharing his screen. Make sure you do a.
Jorge is here. Jorge is going to start.
No, you can start. Go ahead, Cameron. And this is, again, we'll have time for some questions in between. Hopefully this will be interactive. Thanks everybody for joining us.
Okay, can I go? Thank you.
Yeah, go ahead.
Yeah. Thank you, Dr. Friedman and SART team for giving me opportunity to talk in this symposium, very important symposium. And the title of my talk is CARB or CDKN2 IF's role in e-cigarettes and high-fat diet-induced hepatic cardiovascular disease. Can you see my screen clearly?
Yes, we can. It's great.
Okay, thank you. Okay, so I'll divide my talk into 2 parts because I know this symposium mostly focus on cardiac dysfunction, the effect of e-cigarettes on cardiac dysfunction. But here I am going to tell you a little bit about hepatic steatosis or NAFLD, and also try to make a link between liver and disorder with heart dysfunction and the role of CARB in this, whether it can make a bridge between heart and liver or not. So of course this is a very preliminary data I'm going to share with you. So I'll divide, as I told you at the beginning, I'll tell you a little bit about CARB's role in fatty liver disease, and then in the later part, I'll tell something about cardiac dysfunction also. Okay, so NAFLD is a very leading cause of hepatic disease, including steatohepatitis, liver failure, and hepatocellular carcinoma. And almost 1/3 of USA population have chronic NAFLD and highly prevalent in obesity and diabetic people. And it is believed that soon it is going to be common reason for liver transplantation in the USA. And NAFLD also is a risk factor for insulin resistance, metabolic syndromes, and other cardiovascular disease, etc. So ethnically, Hispanic population has highest incidence rate compared to other ethnic groups. And of course, the molecular mechanisms of NAFLD are not still clearly understood yet, and hence therapeutic regimen is yet to be developed. So in this study, our aim is to focus in this area with basic or molecular mechanism NAFLD, and I am trying to show you the role of CARB in the development and progression of NAFLD, especially in the mice. Okay, so next slide. Okay. Okay, so in this study, we use a diet-induced obesity mouse model as a NAFLD model, and here you can see in the figure A, in the left panel, figure A, you can see the liver sections from on the high-fat diet-fed mice have higher accumulation of fat in the liver, and that liver samples, you see the expression of carb significantly decreased in figure B. Carb expression significantly decreased compared to normal so diet control fed mice. And also we treated HepZ2 cell with palmitic acid, and you see in the figure C, the expression of carb significantly decreased in compared to control vehicle cells. So altogether, it suggests that carb, in response to free fatty acid mediated stress or high fat diet mediated stress, carb expression significantly goes down. So it suggests that maybe, but I don't know what is the importance of this down regulation of carb in the metabolic stress. So we propose that maybe the carb can be associated with fat accumulation or the development of fatty liver disease in the mice. So now, in fact, in the right panel, you can see when we knock down carb in the HepZ2 cell, and the triglyceride level significantly increased, is endogenous triglyceride level significantly increased, and and figure B, when we overexpress carb in the hepatocyte or HepZ2 cell, then endogenous triglyceride level significantly decreased. So, suggesting that carb can be a regulator of fat metabolism, or it can maintain triglyceride level in the hepatocyte. And also, next slide, you see here, we want to see also whether carb can regulate the ectopic fat accumulation in hepatocyte or not. So what if here we have treated HepZ2 cell with palmitic acid for 24 hours, and after that you see the fat accumulation or triglyceride level compared to control in carb overexpressing cell significantly decreased. So altogether, it suggests that carb can be associated with fat accumulation or fat metabolism in the hepatocytes. So to understand more details, we did transcriptome analysis by RNA-seq method, and you see many different pathways have been affected in the hepatocytes after carb knockdown. But interestingly, we found that gene associated with ER stress or unfolded protein response pathway accumulated in the carb depleted cells. And you see figure A or B or C, I'm to show you that the genes associated with ER stress and fear significantly altered in the carb knockdown cells. And Western blot also showing you in the figure D, you see GRP8, CHOP, CHOP protein significantly increased after carb knockdown in HepZ2 cells. So maybe the ER stress also, ER stress also involved with fat metabolism and fat accumulation or steatosis is already known. And next, also interestingly, we found that after carb knockdown in HepZ2 cell, the gene associated with PFAR alpha signaling pathway like CPT2, ACAD-M, and ACAD-9, those are involved with fat breakdown or beta oxidation significantly decreased after carb knockdown in HepZ2 cells. On the other hand, the gene associated with triglyceride synthesis, like GPAT, is significantly increased. So it indicates that maybe carb regulate beta oxidation or fat metabolism or PFAR alpha signaling pathway in the hepatocytes. In the right panel, so we try to see whether carb overexpression can rescue fat accumulation in the obese model or not. So we overexpress carb in the obese model by adeno-associated virus gene delivery, and you see in the figure A and C overexpression of carb significantly reduce the fat droplets or triglyceride level in the mice liver. So suggesting that carb can be a protector against the development of fatty liver disease in mice at least. Okay, so now come to the nicotine and high-fat diet, because we know high-fat diet is a risk factor for fatty liver disease, but we know smoking also can be associated with the uh, of the uh, obesity associated uh, disease pathophysiology like cardiovascular disease or insulin resistant diabetes, etc. So our lab has been working in this area a long time, and we want to see whether nicotine and high-fat diet has any combined role in the fatty liver disease or not. In fact, we already published several papers in this area, and recently we have published a paper in hepatology showing that when e-cigarettes are in e-cigarettes and high-fat diet, uh, apoinal mice exposed to e-cigarettes and high-fat diet, then the fatty liver disease severely exaggerated in the ApoE null mice. As you see here, figure compared to figure A and figure B, HN staining, you see the in combination the fatty liver disease exaggerated compared to control. uh, so mechanistically, we found that the circadian system has been significantly altered in combination of nicotine and e-cigarettes and high-fat diet uh, mice. So it suggests that maybe it's already known that circadian system, especially BMAL, in essence of BMAL or the lipid metabolism is uh, altered and accelerated the steatosis in the mice. And another study we already have shown that in wild type mice, the previous experiment we have done in apoinal mice, but in wild type mice also we repeat the similar experiment in wild type, and we found that uh, in combination of nicotine and high-fat diet compared to control e-cigarette 0% or 0% nicotine or saline control, the cardiac function in terms of like uh, LBFS or BCF or LBEF or left ventricular fractioning shortening or left ventricular ejection fraction in terms of this parameter uh, uh, severely in presence of 2.4% nicotine and high-fat diet severely compromised compared to control saline or e-cigarette 0%. So systolic function, specifically systolic function, severely compromised compared to saline or e-cigarette 0% nicotine mice. So and we also found that the in presence of nicotine and high-fat diet, the cardiac function compromise also cardiac. It can correlate with the structural abnormalities in the high-fat diet and nicotine treated mice. Now, now in this figure, I want to show because apoinal mice, we have to come to see whether what is the condition, what is the status of carb expression in this e-cigarette and high-fat diet fed liver. And we found that you see, compared to in figure C, compared to control saline group, the e-cigarette and high-fat diet treated mice have significantly lower expression of carb compared to control. So what is the importance of these lower expression of carb in these mice? So we propose that maybe carb, if you can overexpress carb in these mice, then carb can rescue, maybe can rescue the phenotype. So then in our very recent experiment, we have overexpressed carb in this in this group of mice in this and found that you see overexpression of carb in the liver in fact, we overexpress carb in the liver of these mice, and you see when after overexpression, you see the cardiac function in terms of LBFS or LBEF or BCF. You see compared to control high-fat diet saline group, or is in the e-cigarette group, the cardiac function severely or systolic function severely compromised or reduced. But when you overexpress carb, for example, here you see the cardiac function rescued significantly. Similarly, LBEF also same result. We can see the similar effect, and in terms of BCF also we found almost similar effect. So altogether, we conclude that overexpression of carb in the liver can rescue the heart function. So this is very important observation, maybe, but very preliminary data. Still, we are working on this. Maybe we can figure out more important what is the mechanism how carb overexpression in the liver that rescue steatosis or fatty liver disease, and then rescue also heart function. So we want to make a link with the carb. What is the mechanism here? Our future study will answer this question. Thank you.
Great. We have some time for questions. Anyone have any questions for Dr. Hassan? Do you see any clinical relevance to potentially using CARF for people that are on e-cigarettes or CARF mimetic?
No, because nobody done before anything, and no studies done before, or it is very, very new observation. We don't know.
Right. But do you think it's a potential in the future?
I hope so, because there are, of course, I think there is controversy between whether carb disorder, hepatic disorder can affect heart function. So if we can rescue liver function by carb activation, then it can also rescue heart function. I think very interesting this one.
Okay.
Yeah, I think there are some studies showing the actually 3 axes: liver, heart, muscle. So there is a good possibility.
Yeah, because NAFL is already people suggest that NAFL also very independent risk factor for cardiac dysfunction, but still there are controversy. There are not very strong proof.
This is called actually hepatic metabolic syndrome, hepatic manifestation of metabolic syndrome. So, there is a possibility that you will be able to also restore cardiac function, not only liver.
Yeah.
Great. Okay, we'll move on. Thank you so much, Cameron. Great data, very nice. We'll move on to the next presentation entitled Oxidative Biomarkers and the Assessment of Health Effects Induced by Electronic Cigarettes. Our presenters are Rajat Gupta, who's a PhD student at UCLA, and Jesus Arujo, who's the professor of Medicine and Environmental Health Sciences and Director of Environmental Cardiology, Department of Medicine at David Jeffrey School of Medicine at UCLA. Please upload your slides.
Dr. Friedman, I'm unable to share my screen.
Okay, let me see if I can figure out what the problem is with that. Where did we go? Back to meeting. Not that.
Yeah.
Okay, thank you.
Okay,
Yeah.
good.
You please also make it full screen. Thank you.
There we go.
Great.
Oh, perfect. And thank you for the invitation and for the introduction. And can you hear me?
Yes.
Okay. And Rajat Gupta, and who, as you mentioned, is a second-year graduate student in the molecular toxicology program, is going to present an ongoing work in a clinical study with Dr. Holly Middlecoff, where we have used biomarkers developed as a part of our air pollution work. In those studies, we have used several hyperlipidemic and animal models, such as ApoE knockout and LDL receptor knockout. Let me see if I can use the pointer. And normal lipidemic C57 black 6. We expose them to various air pollutants, like in ongoing studies at our air pollution inhalation facility at UCLA, and we measure various endpoints. We have been very interested in translating these detected effects in mice into humans to make sure that those effects do occur in real life and human biology. Then findings in the human studies can then be tested in mice for an optimal crosstalk in between both types of studies. And as you will see, in the case of the electronic cigarette studies, we have actually started with the humans first and then gone to the animal work and for further characterization. Let me share a useful perspective from the air pollution studies, where it's been quite difficult to dissect and elucidate pro-oxidative and pro-inflammatory effects and leading to worsening atherosclerosis. It wasn't until we tested ApoE knockout animals and who were exposed to diesel exhaust versus filtered air for a couple of weeks and tested on oxidative products of polyunsaturated fatty acids that can be seen in these schematics. Products of the linoleic acid, which is an carbon 18 and 2 double bonds, called the HOATs, or products of the arachidonic acid, which is 20 carbons and 4 double bonds and called the hits. And we detected those products and that increased from 2 to 4 folds in the blood of the animals exposed, as well as in several other tissues. Human studies allow us to validate those findings in mice with an experimental design that allows us to determine what will be the effects after several weeks of exposures. So we tested in the blood of students who traveled from UCLA to Peking University in Beijing and then back in Los Angeles. Blood and urine samples were taken before at Beijing as well as in LA. And those same products that we had seen elevated in the case of the mice were markedly elevated also in the students who had traveled to Beijing 6 to 8 weeks after arriving there. And the levels returned to baseline when back in Los Angeles. What Rayat is going to show you now is the usefulness of these markers in detecting pro-oxidative effects, if those were present, in the case of the electronic cigarette studies.
Hey, so just an introduction to, I think it's also going to be in some of the other talks. Can you explain why you chose ApoE knockout mice?
We're very interested to evaluate the atherosclerotic effects of the air pollution. So we certainly needed to have an animal model that it will be resembling, atherosclerosis. We also tried to determine whether the same effects could be obtained in normal lipidemic animals, and certainly the effects were not well as easily detected or identified as when we did it in the ApoE knockout. So this is something that it was like a more significantly determined again in the pro-atherosclerotic mouse, and it's one of the concerns that we had when we moved when we're trying to translate the mouse studies into the humans is, will we be able to determine these same effects in normal lipidemic or in young healthy subjects with totally normal levels of the lipids? And I have to say that we had failed in some previous attempts but not because of the types of the subjects, but because of the types of the studies. So in the air pollution studies, we cannot expose for more than 2 hours. And the animals were being exposed for several weeks. So how could you really reproduce the effects? So it was with a design where we actually tested on the effects 6 to 8 weeks after arriving to Beijing, that we were surprised to see that exactly the same levels that were being induced in the hyperlipidemic model were also being induced in subjects who were totally normal. Thank you.
Great, thank you.
Good afternoon, everyone. My name is Rajat Gupta. I would like to first thank Dr. Friedman and the organizers for giving me this opportunity to present today at the symposium. I'm very excited to share some of my recent work on the use of oxidative biomarkers in assessing the health effects induced by electronic cigarettes. The work that I'll be showing you today is currently unpublished and has been submitted for peer review. Before I show my data, I would just like to briefly mention that the work in this project was recently presented in the form of a virtual poster at the American Heart Association Scientific Sessions meeting last year. So with the increase in the electronic cigarette use, especially among the middle and the high school students, there is a very big concern about whether electronic cigarettes can induce cardiovascular risk. We have known from epidemiological studies and for a few decades now that TC smoking or tobacco cigarette smoking can increase cardiovascular risk and is also regarded as one of the single most preventable cause of early death in the United States. Electronic cigarettes were introduced in the market as a safer alternative to these conventional cigarettes. However, the long-term use of electronic cigarettes in inducing cardiovascular risk is currently unknown, and that is going to be the focus of my today's talk. One of the very early implications of cardiovascular risk and oxidative stress in habitual electronic cigarette users came from this study published in JAMA Cardiology in 2017 from Dr. Middlecoff's lab and in collaboration with our lab, showing that the electronic cigarette users had differences in the heart rate variability frequency components, as you can see from panels A, B, and C. This data suggests that there was a shift in the cardiac autonomic balance towards a sympathetic predominance in the EC users as compared to control participants. More importantly, this study also showed that electronic cigarette users had increased susceptibility of the low-density lipoprotein to undergo oxidation in comparison to non-users, suggesting that a potential for increase in oxidative stress. Based on this study, we asked two very fundamental questions. Does chronic electronic cigarette use increases lipid peroxidation and or impair antioxidant activity? And the second question, can chronic electronic cigarette use increase cardiovascular risk in healthy young adults as compared to the previously already known cardiovascular risk in chronic TC smokers? Therefore, we designed a cross-sectional study and we recruited subjects in each of these three different groups. They were either non-users or control subjects, or electronic cigarette users or tobacco cigarette smokers. The subjects in the EC users and the TC smokers group either used an electronic cigarette or smoked a tobacco cigarette for at least 1 year. We collected the blood at baseline from each of these different subjects and harvested the plasma to measure various parameters. First, we were interested in looking at the oxidized levels of the linoleic and arachidonic acid by mass spectrometry. And as Dr. Arujo previously mentioned, that these oxidative products served as biomarkers in our air pollution studies, where we have seen the increased levels in response to air pollution. We were then interested to also look at the peroxidase 1 antioxidant enzyme activity in the plasma, and this is associated with the HDL. Then we were interested to look at the total antioxidant capacity, which represents the sum total of all the antioxidants in the plasma. We then looked at glutathione metabolism and finally cardiovascular risk. The hypothesis of my study was that chronic use of electronic cigarettes for greater than 1 year induced lipid peroxidation as well as cardiovascular risk. The first result I'm showing you here is how does chronic use of EC and TC affect oxidized products of linoleic acid. And in this study, we measured particularly the lipoxygenase-mediated oxidized products of linoleic acid, which are the 9-hode and the 13-hode. Surprisingly, if you look at the total hodes, which is the sum total of 9 and 13 hodes, there was in fact a decrease in the levels in between EC users and TC smokers, suggesting a decrease in lipid peroxidation. We then looked at the levels of the oxidized products of arachidonic acid, and particularly we measured the levels of these 5 heats, 12 heats, and 15 heats. And we actually noticed that there was no change in any of these 3 different groups. We then looked at the levels of the parental substrates of these oxidized products, which are the linoleic acid and the arachidonic acid. And as you can see here, ECUs and TC smoking differentially regulated their levels. EC users in particular showed decreased levels of the linoleic acid and the arachidonic acid, whereas TC smokers had increased levels of the arachidonic acid only in comparison to the non-users. This data actually suggested that the ECUs or TC smoking can affect lipid metabolism. While we did not observe any differences in the peroxidase 1 antioxidant enzyme activity that would explain any of these effects, we in fact looked at the levels of the total plasma antioxidant capacity. We measured this using a biochemical assay, which actually measures the inhibition of the oxidation of this compound ABTS to form this oxidized product. And interestingly, we noticed that both EC users and TC smokers had increased levels of the total antioxidant capacity as compared to the non-users. This clearly suggested that there is an increased compensatory antioxidant defense due to the effects of smoking or EC use. This was actually consistent with another previous study showing that in case of healthy young smokers, there was an increase in the total antioxidant capacity, suggesting that the antioxidant defense was greater that could combat the pro-oxidative effects of smoking. This was a very interesting data to us, and we further looked into the mechanisms of the antioxidant defense, and that pushed us toward looking at the glutathione metabolism. As we know, glutathione is a potent cellular antioxidant, and it can circulate between the reduced and the oxidized forms in response to oxidative stress or detoxification. So here we looked at the levels of the various glutathione parameters, and we saw some changes. First, I'm showing you the levels of total glutathione. We saw that it was significantly higher in case of EC users, but only showed an increasing trend in case of TC smokers, which was not statistically significant. In the case of reduced glutathione, it was the opposite. We actually saw an increase in both EC users and TC smokers, which was statistically significant only in case of TC smokers, but only borderline significant in the case of EC users. The oxidized glutathione there was no difference in the TC smokers, but there was only a trend of increase in the EC users. More importantly, when you look at the ratio of the reduced to the oxidized glutathione, there was in fact no change in the EC users, but there was a significant increase in the TC smokers. And this was important because you can see that in case of EC users, we think that there was a redox homeostatic balance, which was not present in the case of TC smokers. We used this to ask the question whether this ratio can be used as a marker for cardiovascular risk in healthy young adults. And it turns out that we can use this based on a previous study by Ashwak et al. Published in the Journal of American College of Cardiology in 2006. They actually showed that there was a significant association between the levels of this glutathione redox with the carotid intima media thickness. This was positively correlated. The study also showed that this ratio was significantly associated with the Framingham risk score that predicts the 10-year cardiovascular risk of an individual. This study showed that the glutathione redox status can predict the early atherosclerosis in a cohort of non-smoking healthy individuals. We use this to our advantage and to determine whether the ratio that we are seeing in our case, whether we can predict anything about the cardiovascular risk. And it turns out that, as I showed you in the previous slide, we only saw an increase in the ratio in the TC smokers and is in comparison to the non-users, which showed that TC smoking, in fact, increased cardiovascular risk. Lastly, we did a multivariable analysis to identify when we combine all the biomarkers in our study. We wanted to use statistical modeling to identify whether EC users can be segregated from non-users and TC smokers. So we used 2 different models. First one on the left, I'm showing a classification tree where you can see that based on these two biomarkers on the top, the 13-hode by linoleic acid and the 9-hode, we can see that the EC users can actually be segregated from non-users and TC smokers with an accuracy of 73.9%. In another model, this was also another model where we used all the biomarkers together. This is the nominal logistic plot model. And in this model, we noticed that while non-users were segregated on the bottom left, TC smokers on the upper right, EC users were in fact in the middle. and the prediction accuracy of this model was 94.4%. This data suggests that the effects of ECUs were intermediate between non-users and TC smokers. Therefore, in the overall summary, I showed you the data that the TC smokers and EC users had some common effects as well as some differential effects. Some common effects were that both of them had reduced levels of total HODs, increased total antioxidant capacity, while no changes in the levels of total heats or PON1 activity. Some of the differential effects were that TC smokers had increased levels of arachidonic acid, as well as an increase in the ratio of reduced to oxidized glutathione, indicating increased cardiovascular risk. On the other hand, EC users had lower levels of the arachidonic acid and linoleic acid levels. More importantly, using multivariable analysis, we saw that EC users can be segregated from non-users and TC smokers, suggesting a potential increase in cardiovascular risk. With that, I would like to acknowledge the support of Dr. Jesus Arujo and all the lab members in Dr. Jesus Arujo's lab, as well as our collaborators and the funding sources. I'll be happy to take any other questions. Thank you.
I was muted. Hey, I'm sorry. Can you go back to your conclusion slide? Rajat?
Yes.
I was muted.
Yeah,
So
sorry.
probably you didn't hear my question.
Right. So what do you consider bad and what do you consider detrimental? What do you consider beneficial? And would you say that conventional cigarettes are worse than e-cigarettes? You're sort of implying this on that bottom.
Yes, so it was very interesting in our study that we actually were initially, when we started the study, we were expecting to see an increase in lipid peroxidation. That would actually mean that there is increased oxidative stress and increase in cardiovascular risk, which was already currently known in the case of EC users. But what was really interesting in our study is that we saw the opposite. We saw that in the case of healthy young adults with no known comorbidities, there was in fact an antioxidant defense that was actually working to their advantage to combat the effects of this smoking. So yeah, in case, but we also noticed that with the multivariable analysis, we also saw that there was the segregation, and we looked at some other markers also, which showed that TC smokers in fact are actually showing effects that are not good. But in case of biomarkers, I would tell you that they're actually showing the opposite to what we would expect.
So which would you say is worse from your data, the EC users or the TC users, TC smokers?
So I would say that TC smokers are definitely bad according to my data, because we are also seeing a significant increase in the glutathione redox.
Yes, oxidative stress.
Oxidative stress, yeah.
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 Right. So now my question.
That you don't see in e-cig users.
Correct. Right. So you can say that there is something really bad happening in case of TC smokers because of the effects of TC smoking, that the body is actually overcompensating the effects on glutathione metabolism, which was not the case in the case of ECUs.
Mm-hmm.
Okay, so now I have a question. In your case, because you used normal, healthy subjects in all three groups, what will be your prediction that many of them are also obese? So you are exposing them to stressors like e-cigarette or cigarette smoke, as well as obesity.
I see. That's a very good question, actually. So when we recruit subjects in our study, we make sure that there is actually no underlying condition. So there shouldn't be any like an obesity that is to a level where it is considered a disease. So these were supposed to be like perfectly normal, healthy subjects. But we didn't actually, in the study, we didn't see an effect of anything additional to just being anything with normal, like no other conditions can actually, have an effect here, because we didn't study any of that, because the subjects should apparently not have any of those conditions.
Yeah.
Were any of them obese, or were they all normal BMI?
They were all normal BMI.
Yeah.
We had a strict screening criteria to exclude subjects with any of the, which didn't pass our qualifying criteria.
Yeah, my prediction will be that if your subjects are also obese, you will see detrimental effects of both e-cigarettes as well as conventional cigarettes.
I see. Yeah. That's possible. That's quite true, actually. Yeah, it could be. It would be definitely the case. With some underlying conditions, there could be definitely an effect with the EC smoking and the EC use and the TC smoking. Yeah.
But your results are, I mean, the findings are really, outstanding, and you can predict also. Okay, in terms of oxidative stress, conventional cigarette smoke, they have a lot of other things besides the nicotine, and for that reason, e-cigarettes are much more refined. But as long as they are normal, healthy person, it will have no effect. But with other comorbidities, it will have effect on, definitely based on our animal experimentation on liver, heart, as well as skeletal muscle.
Yeah, I agree.
I think that is, it is quite possible what you're saying, that if you have other comorbidities or you have other risk factors, that the compensatory responses the individuals are having are not enough, are not sufficient to counteract the harmful effects of the smoking. So in the case of these healthy human subjects, you can see that there must have been initially pro-oxidative effects that if we had looked earlier, let's say at a few weeks after, like how we did in the air pollution studies, a few weeks after they started smoking. So you could be seeing these increases in the oxidation. That would be a prediction, and increases in the heats, increases in the HOTs and all that. That will be confirming the hypothesis of this increase in the lipid peroxidation. It has been shown also by other groups and with the erasoprosteins with short terms of smoking. But as we evaluated much longer, like after a year of smoking, that there are all these compensatory responses that result in increasing the total antioxidant capacity that happen in both cases. So there is a very discrete difference in between the smokers and the electronic cigarette users. In the smokers, the increase in the total antioxidant capacity was not sufficient to normalize the levels in the glutathione metabolism. So that's what Rajat is mentioning. It appears to be almost like it is like an overcompensation. And if you think about it, when thinking about it, why would the body overcompensate on something? Why would you try to do actually better or what you usually do at a baseline and when you are in completely homeostatic state? It doesn't make any sense. It's gotta be that these pro-oxidant effects persist, and that's why you have this response that's just trying to counteract that, but it doesn't do it. You're gonna do it entirely, and that's why you still see an effect in the glutathione ratio. But in the case of the electronic cigarette user, it's different. The increase in the total antioxidant capacity was enough to counteract those pro-oxidative effects of the electronic user. And when you look at the glutathione, which is the best estimator really of redox states, you don't see changes. You do see changes in the reduced glutathione or the oxidized glutathione, but when you look at the ratio, there are no differences. So that's, I think, that what Rajat is referring to. It appears that this is indicating like a homeostatic balance. Now, what will happen in the obese, as you said, or in diabetic? Maybe the obese or the diabetic will not have that ability of reaching that homeostatic state. And if that is the case, so it may resemble more of what is happening with the tobacco cigarette smoker. And in which case, maybe in that subpopulation, you will still be seeing the increase in the cardiovascular risk that you will be seeing with the smoker, but maybe it's not as noticeable in the case of the EC user. I mean, that's one way of interpreting the data,
In our animal experimentation study, we did quite a few, and at every time ratio of reduced GSH to oxidized GSH, which is much lower, both by high-fat diet or by nicotine. But if you combine these two together, then it's markedly lower compared to either insult, suggesting that it causes a high level of hepatic oxidative stress or cardiac oxidative stress.
It makes sense, right? Yeah. So I guess that you're summing up like various either call it a risk factors or it's when you have like some sort of interaction, right? That whether it is an environment interaction or gene environment interaction. Yeah, it makes sense.
Thank you. Very nice data.
Thank
Very
you.
nice study.
Are you doing any work in rodents with this?
You're asking me?
So,
Yeah, you or your boss?
well, I think that this is leading to the next
Yes.
presentation.
Okay, go ahead. Okay, thank you, Rajat.
Thank you. Thank you, Dr. Friedman.
Thank you so much. It's beautiful.
Yeah,
Oh, I have
great.
one more question. You haven't done any dual users? I thought Amia would ask that question.
Right.
You haven't done any dual users in this experiment, right?
No, they were either non-user, they were either electronic cigarette users or tobacco cigarette smokers, but
Yeah,
not
it's very simple. Yeah.
Right. So maybe it'll be interesting to look at the dual users next time.
But that there was I don't know, am I unmute? Yeah. But there was a very astute of comment and from a reviewer of his manuscript who was asking, some of these subjects had been smokers in the past. They quit smoking and now they're pure electronic cigarette user, supposedly, right? And so then they say, could it be that actually some of the effects that you're seeing in the electronic cigarette user is actually a residual effect from their years-old habit of the tobacco smoke, or is this really something that is due to electronic cigarette user? So Rajat did a nice subgroup analysis. And while he cannot rule out that there is some residual effects that is actually happening years after a person has stopped smoking, clearly some of the effects had no relation with that, and they were clearly induced by just pure electronic cigarette use.
Okay, great.
It's a very good question and very good.
Thank you. Okay, we go on to the next talk is investigating HRV modulations during acute electronic cigarette exposure in C57BL6 mice by Jocelyn, Castellanos, and Jesus Arujo. Go ahead, Jesus and Jocelyn.
Yes, I think that Jocelyn is going to share her screen.
I'm probably not doing a good job of pronouncing your last name. Arujo?
I know that the AU combination is a killing combination in English. Araujo.
Arujo, okay.
But it's there. It's almost there.
Arujo.
Okay, yeah. So this presentation and this work and that will be presented by Jocelyn. As you mentioned, she was a master's student in environmental health sciences and in our School of Public Health, and she started this work and actually as a part of her master in our lab, and she completed it, and now she's a associate researcher in the lab and is in charge of a good portion of the electronic cigarette work in our lab. So let me go back to that first slide that I showed and in the previous presentation, which is like this interest that we have in trying to integrate the animal and the human studies, which has been a predominant theme in our air pollution work, and has also, in a way, led our efforts in the electronic cigarette work, and which has been clearly more recent. But very interestingly, we actually started in the opposite direction. In the case of the air pollution, so we started with the animal work, and then we identified effects, and then those effects that then were translated in the case of the humans. In the case of the electronic cigarettes, I have to say that I was very dubious initially that the electronic cigarettes could be causing any sizable or meaningful effect, and which was perhaps a more, or had been identified as a clear effect in the case of the air pollution. And then we actually conducted human studies, and these were clinical studies and led by Dr. holy middle calf, and wanting to identify whether was there something in there, was there something that really justified like also developing all the tools and systems and models and so on required like for animal work. So in this case, we actually started with the human studies, and once we obtained like some data from the human studies that were actually quite interesting, so we decided to move into the animal studies. And Jocelyn will be showing like one of the things that that really triggered our attention. It was the findings and that the human studies that show on effects on the heart rate variability. So that has been our initial interest in determining whether those same effects that have been identified in humans can also be reproduced in the animals. And we also did something different than with air pollution study. We started with susceptible animals, as you mentioned, as you asked, and Theo, why did we use Apoinocar animals? So actually, in the case of the mouse studies, because we started like coming after the human study, we started more with C57 Black 6 animals, and we'll be moving into hyperlipidemic animals after. And the idea is like with the data and the intake that we obtain in the mouse study, so we can come back and refine also the designs and of our human studies, and we can have like a good integration in between both. So this led into the project that we're currently conducting, funded by the TRDRP, that allow us to establish our mouse facility for electronic cigarette exposures at UCLA that we call MASER, or MASER, Mouse Easy Exposure Resource at UCLA. And that has been a joint effort, and thanks to Ken Roos, and who is the director of the physiology core, and where we actually have physically located the machines. And there is a very, there's a logistical reason why that happened, since our main interest had actually been at looking at electrophysiological effects and effects on the heart rate variability. And he actually has a full setup of his cardiac telemetry and HRP studies that can perform in his core. And we use the same chambers that you're using and developed by Max and Chao, and partnered with an Holy Middle Calf and Yifan Su, who is an air pollution and aerosol expert, and in the air pollution also doing significant work in the electronic cigarette arena. And one of the things that it was challenging and from the beginning is that we wanted to do something different with the animals as it's been done with the humans. So with the humans in general, the way how it's been done is that the patients are recruited and they get exposed to the easy, they use the electronic cigarette, and then an electronic and electrocardiographic tracings are obtained after, and then we do all the measurements and to determine whether there have been changes or not. We actually wanted to see in real time whether the exposure will be causing those same effects exactly at the same time as we were conducting the exposures. But we had a logistical problem, and is that it was difficult really to insert the telemetry antenna that it was required. And you can see that there were some changes in the configuration of the chambers that, thanks to Joe and Cortez and Max Chao, we were able to do. So we were able to actually place the telemetry antenna just underneath the exposure chamber. And that allows us that when the animal is being exposed, so all these signals are being transmitted and are being captured, and everything is being recorded. So now we can say whether things are starting within a matter of minutes, and we don't have to wait for the whole 30 minutes or 60 minutes, and which is what it will take for the session of the exposure to the electronic cigarette. So without further ado, so let me just now give this to Jocelyn, who will continue and will present the work that she's been doing.
Hello, everybody. Sorry about that. I was having trouble unmuting my mic here. So yeah, as Dr. Arujo mentioned, my name is Jocelyn. Thank you for the invitation. I look forward to sharing some of the work that I've done during my master's and now as a SRA at Dr. Arujo's lab. So briefly, this slide or this figure was published in Dr. Mitochondrial's study or review in 2019. And just as an emphasis of what Dr. Arujo was saying, she showed certain mechanisms that would increase cardiovascular risk with electronic cigarette use. And the one that we're going to focus on today is heart rate variability. So based on previous human data in which chronic e-cigarette users exhibited measures suggestive of decreased heart rate variability, there's a potential for an increased risk of there's a potential for increased cardiovascular risk with EC use, looking at this or using this diagnostic method. And so before we kind of launch into the study, I really briefly wanted to go over what heart rate variability is. Now, heart rate variability was developed as a non-invasive diagnostic method that's used to assess autonomic health and determine cardiovascular prognosis. In general, high variability implies good cardiovascular health and a well-functioning autonomic control mechanism, whereas in low variability is indicative of bad cardiovascular health, and it's associated with insufficient adaptability of the autonomic nervous system to maintain homeostasis with respect to heart rate. So overall, changes in autonomic regulation of the heart can have major implications associated with arrhythmias and acute ischemia, among other cardiovascular diseases. But two ways that you can actually analyze heart rate variability is through the time domain and the frequency domain. Now, the time domain quantifies variability between beat-to-beat data obtained from RR interval series and is influenced by changes in both the sympathetic and parasympathetic activity. So you can think of the time domain as the amount of time in between heartbeats. And really quickly, just wanted to show you an ECG graph so you can follow along with me. On the other hand, we have the frequency domain. So because the sympathetic and parasympathetic branches of the autonomic nervous system are strongly influenced by heart rate, there is a difference in the speed by which heart rate changes can occur. So given the differences of speed responses, you can think of the frequency domain in terms of how the RR intervals are being modulated with respect to sympathetic and parasympathetic contributions. And so in this study, we really wanted to focus on this centralized question, which was, can e-cigarettes decrease heart rate variability during hyperacute exposure in mice? And I want to underline two words here, during and hyperacute. So there's actually been previous human data as well as mice data that's just recently been published of the effects of heart rate variability decreasing or being modulated by exposure to e-cigarettes. But these exposures have only occurred in an acute or even a subacute setting, as well as potentially a chronic setting. And this is only after these e-cigarette exposures have happened. With our study setup, we're actually able to record all of this information in real time. And so this information is really giving us what's happening during this exposure. So really briefly, our study design, we first implanted these 12-week-old male mice in the C57BL6 background with these radiotelemetry devices and a lead-2 arrangement. And then our study protocol or exposure protocol is a little bit lengthy, but we first wanted to acclimatize our mice in these chambers that Dr. Xiao and Joe have designed. And we did that for 1 hour. And this was to decrease their or adjust for any increased heart rate when we were moving the mice from their cages to their chamber. We first expose into our air, which is our primary control. And in order to incorporate real life conditions, we actually included a post-air or a post-exposure event. Humans don't necessarily smoke over very long periods of time. They do it in suctions, or that's what we've seen. And so based on the literature, we actually, and based on recommendations by Dr. Sham, we included just 15 minutes, or we included these post airs to last 45 minutes, and our air to last 15 minutes. And we did this to replicate what we were doing for our e-cigarette exposures. So we included 2 e-cigarettes exposures, and mice were exposed to a 2.4% classic tobacco Blue Plus cigarette. So over that 15 minutes, they had 4-second puffs per 26 seconds of air. And lastly, we actually incorporated a secondary control, and this was our PBS. And so previous studies associated with these rodent chambers actually used aerosolizing as their control. And so we kind of wanted to have a similar way to make our study a little bit more comparable to what others had used. And so we actually ended up using a PBS. And each of the exposures, of course, had their own post-exposure event. And lastly, for our HIV analysis, we collected electrocardiogram, temperature, and activity in real time, and then later analyzed particular heart rate variability parameters. The heart rate variability parameters that we looked at were really the SCNN and the RMSSD. And I want to give a little bit of a disclaimer. This is unpublished work, so we're going to show limited data here. But the SCNN is the standard deviation of the normalized RR intervals. It indicates total autonomic variability, or the variation within both the sympathetic and parasympathetic branches. However, in short-term recordings like ours, the primary source of that variability is parasympathetic mediated. We also looked at the RMSSD, which is the root mean square of successive normalized RR interval differences. So it really indicates short-term variability, and it's thought to reflect parasympathetic response. Our data, so we actually use a linear regression model. And so with no real differences between air and post-air exposure, we actually combine those. And to see the overall effects of e-cigarettes, we actually pull that together. And so here we have a comparison between pooled EC and combined air. And so when comparing these two, pooled EC and combined air, the e-cigarette exposure significantly decreased the SCNN and the RMSSD by 5 and 9.3 units respectively. In conjunction, compared to our secondary control, which was the PBS, it actually resulted in even greater reductions in both of those parameters. And so therefore, reductions in these components indicate a decrease in parasympathetic activation, and actually suggest acute e-cigarette exposures can actually decrease heart rate variability. And so based on this information, we actually wanted to look at, okay, what are they being exposed to in our protocol? What are the aerosol concentrations here? And so really briefly, the methods, we actually collected, or we measured particle number concentrations and particle mass concentrations at 1 second intervals, and this was also in real time. We looked at particle size distributions, and then we merged that information together using this data merge software module. Yeah. And so in our data here, we have here on our left-hand side, I think I can, here we go. So for particle number distribution, we have our e-cigarette aerosol, our background air, and also our PBS. So we can see that with e-cigarette aerosol, there is a bimodal distribution. So there's 2 humps that you can see here almost. And what you can see is that this particle distribution is really concentrated in the fine and the ultrafine ranges. Our air, you can see, is completely zero. So That's nice. But our PBS is actually showed also a bimodal distribution, a little bit lower than the e-cigarette aerosol, but that was a little bit interesting to see, as we thought that it would be a lot lower, more comparable to air. And we'll look at that a little bit more in detail. In terms of particle mass distribution, we're seeing sort of similar things where there's an increase in the e-cigarette aerosol, but our PBS and our background are definitely lower. So as we kind of focus into this PBS, or into our controls, we can see that this PBS is actually a little bit more variable and definitely a little bit higher than our air control. And it actually seems to show similar particle number mass distributions to EC, but again, an order of magnitude lower in both particle number and mass. So with this control established here, this gives us or suggests that these results that we're seeing, the effects that we're seeing, excuse me, that are induced by EC are not due to the mere presence of particles that can stimulate receptors and modulate the autonomic nervous system, but instead these changes are likely due to compositional differences. And so with PBS, we have an isotonic salt solution. With ECs, we have a variation of chemical compositions. And so overall, this study shows that acute or hyperacute e-cigarette exposures can decrease autonomic variability in mice as measured by heart rate variability, and actually demonstrates that e-cigarettes may not be as safe as previously assumed, with implications for cardiovascular health. I want to thank our lab, as well as Dr. Zhu's lab, who did a lot of the aerosol work with us. I want to thank the Mouse Physiology Core, which includes Dr. Bruce, Dr. Jordan, as well as Carla, who did some of this work as well in our lab, and Dr. Shao. So yeah, if you guys have any questions, thank you.
Thank you very much. Beautiful.
I may ask a question to ourselves. How about that?
Sure.
Jocelyn, what do you think that could have been the usefulness of employing that and PBS and aerosol as a control, and versus what is usually done in this type of studies, where simply the electronic cigarette and aerosol is compared against a filtered air or plain air?
Mm-hmm. Well, I think with the filtered air, well, if we look at e-cigarettes, right, they're aerosols. With filtered air, it's in a gas phase without that liquid particle. So with the PBS and the introduction of the PBS, you sort of get that similar feel of it's an aerosol, I would say. And I think that's the usefulness of including the PBS as our secondary control, as opposed to just the filtered air. Yeah.
Do you want to conjecture about some clinical relevance of your work?
Can you repeat that?
Do you want to conjecture about some clinical relevance of your work? What does this mean to people that use e-cigarettes?
Sure, I'll take a stab at it.
Okay.
So I think,
I have a question.
oh, did somebody say something? I'm sorry.
Oh, that was me. I can wait you answer, Dr. Freeman. I had a quick question. Well, no.
Well, let her answer my
okay,
question,
you know,
Renee. Let her answer my question.
Sorry. Okay.
Okay. I think some of the, I guess the clinical conjecture, I think it's hard to say at this moment, particularly because there's other data that we didn't show, particularly in the post-exposure, that shows a decrease, so almost back to normal. So I think over a longer period of time, not just in the hyperacute, but more in the chronic sense, you'll see a buildup of more decrease in heart rate variability that could lead to potentially, arrhythmias or arrhythmias or cardiovascular disease in the ischemic character.
Great. Renee, what was your question?
Okay, go ahead.
Jesus, do you want to answer also the question, or Renee, did you have a question?
Oh yeah, well.
Oh, I was muted. Okay, I'll wait.
Go ahead, Renee.
Okay, my question was with the aerosol ties with the air that got conditioned, and they were using that more as like a control versus the filtered air. But wouldn't those two airs be like a little different? Would there be more moisture in one or more, you know what I mean? Aerosol, I'm just wondering. And how did you guys do that? I'm just curious. That was it.
Yeah, so yes, there would be more moisture, although we didn't record the relative humidity inside the chamber. I think we just expected a lot lower concentrations, especially for the particle number concentrations. And so that was interesting to see. But the way that we actually did it was we took the chamber itself, and I think I can show you, if I go all the way back in the video, right down here. So we took this entire chamber, we repeated our exposure protocol exactly the same without the mice in there. And if you see this portion down here, where it's connected to a HEPA filter, we actually took a tube and connected that to our various instruments that we use to measure aerosols. Yeah.
All right, not bad. Goodness, thank you.
Yeah, no problem.
Hey, Jocelyn, and hey, Dr. Araujo. I actually had a question. When you were looking at the particle number concentration and your particle mass distribution, I noticed it was more like focused around particles around 0.1. So is there like a higher risk factor for PM 0.1 rather than PM 2.5? Because I know with e-cigs, like a lot of the literature I've read, it's centered around PM 2.5 rather than PM 0.1. I was just curious.
I don't think I am quite at liberty to say that as of yet. There's more of that in the unpublished work that we're doing. So yeah, check back in with me for that.
Got you. Anyway, great work, guys.
But it's a good question, Sam. Which I think that we know very little of what within the electronic cigarette and aerosol is responsible for effects. Just in the same way that we still know very little of what within the air pollution particles are actually responsible for the effect. And in both cases, so we have a solute or solid, particulate component. But in both cases, so you have like a substance that is going between the solid phase and the gas phase. A difference may be also that in one of the cases, you could actually be even affecting, as I heard, humidity and temperature. In the easy aerosols, you have like a heating of the particles. So one of the questions that we labor from the beginning is that what is a proper control to use in these exposures? Because if we compare against filtered air, so certainly we can say, whatever is in the easy aerosol should be causing the effects, but we don't know what. And we don't know if it is a particle, if it is a gas, if it is a volatile or semi-volatile compound in between. We don't know if it has to do with the temperature or what, right? it is very difficult. So this is one of the reasons why, in a way, by serendipity, I think that the PBS and aerosol control was introduced. And it does give a very interesting finding, which is that the aerosol first shows like these very tiny, teeny particles that are much smaller than the electronic cigarette and particles. And obviously a very different composition, right? In the PBS, it's just salt, that's it. And in this aerosol, you have all these myriad of different compounds with a different temperature. If the responsible for these effects is just the physical presence of a particle, even if it is smaller in size or it is smaller in magnitude, shouldn't we be seeing something similar, perhaps in a different degree? Well, on the opposite, we actually saw exactly the opposite. The PBS aerosol not only didn't do anything, the PBS actually changed things in the opposite direction as the EC aerosol. So the PBS aerosol increased the heart rate variability. So when you compare like that, when Jocelyn showed those comparisons between the PBS and the EC aerosol, so those differences are actually much bigger than when you compare the EC aerosol versus the control. So that tells you that just a particle per se is not really doing this effect. Now, then you're talking about the size, and this is an incredibly complicated question. Even in the air pollution arena, oh my goodness, we have all the epidemiological data that is showing that PM2.5 is what is responsible for cardiovascular deaths and metabolic effects and cerebrovascular disease and all that. However, you have all the animal data that is showing that its particles more than 100 nanometers. I mean, the CP0.1, right? So why do you have this discordancy? Well, I think that it's just very difficult to measure in epidemiological data to what actually we're able to measure with the animal world. It could still be that the smaller particles are more toxic, but we're not able to capture that toxicity in the clinical studies. Imagine that, now translate that into the electronic cigarettes, where we don't even have a notion yet of whether particle presence per se or particle size has any role. At least, the data will provoke some questions, that we should be looking at this, and maybe the particle per se could be having a role, but I think that it's important the composition, what is within the particle, not just the presence of the particle per se. But really good question. I think there's more to talk about, certainly.
That makes sense. Thank you.
I guess this is for Jesus and maybe Ken. Do you have plans to look at long-term treatment and heart rate availability, or do you want to collaborate with us who are treating the animals long-term? And do you think that would be interesting to measure heart rate availability and chronic exposure?
If Ken allows me, I think that both things, and we'll be happy to collaborate, certainly.
Okay.
And we do have, I think it's important. There was a recent paper published that it showed that after several weeks, there was also an increase in the heart rate, a decrease in the heart rate variability. But that was when measuring the HRV after the exposures and measure like 4 hours after the exposure. So similar of what also we had reported with the humans. In the humans, we reported like a few minutes after the 30 minutes of exposure. And in the case of the animals, it was like 4 hours after. It's significant. I apparently started seeing this like at week 6 or 7. I think that characterizing this in more details in animals and seeing in the different animal models of susceptibility will make a lot of sense.
Great.
Yeah, so it's something that certainly we could partner.
Okay. Before we go on to our last talk, I want to have 2 announcements. Can you unshare your screen, Jocelyn?
Mm-hmm.
Okay, thanks. So our next SART Institute is, can you see my screen?
Yeah.
Okay, and the next SART Institute is the 4th one, entitled Clinical Epidemiology and Behavioral Skill Development. It's April 16th, 2021. And
Okay.
our next e-cigarette symposium is May 7th, I think it is. So that's, here it is, May 7th. And that will be, announcements will come out for soliciting talks for that one. So our last talk today is entitled Asipimox Prevents Cardiac Dysfunction Induced by Electronic Cigarettes in Mice. Jorge Espinosa Dorado is presenting. Thank you. Jorge, go ahead.
Okay. Okay.
Great.
Okay. Hello, everybody.
Jorge, could you please get the full screen?
Okay, full screen.
Right. Go this one.
There we go.
Thank you. Thank you.
Let me see. Okay, let me second.
No, it's back again.
Okay. Okay, now. Okay, has hello. Our lab have been for a couple of years already interpreting the effect of electronic cigarette in the heart. And as everybody knows, should we know here, electronic cigarette had nicotine. And it has been shown that nicotine produces lipolysis from adipocytes, leading to the increase of glycerol and free fatty acid in the blood. And when free fatty acids are likely to be one of the key elements of ectopic lipid accumulation and lipotoxicity and mitochondrial dysfunction in the heart. We know that the high level of free fatty acids are correlated with an increase of risk of cardiac dysfunction and arteriosclerosis. As well, we know that the elevated plasma free fatty acid level can enhance oxidative stress, leading to vascular endothelial dysfunction and inflammation. High level of free fatty acid can induce a change in oxidative metabolism in several target tissues, as skeletal muscle, liver, and heart. So before we have described a model where electronic cigarette induce cardiac dysfunction. So in this published work, we expose mice to electronic cigarette for 12 weeks. And we expose mice to electronic cigarette with saline, with the electronic cigarette without nicotine, and electronic cigarette with nicotine. And we see that the mice exposed to electronic cigarette have increased level of free fatty acid chronically in the blood. As well, when we take a piece of the heart and we measure the marker of oxidative stress, we see that the mice exposed to electronic cigarette with nicotine have an increase of oxidative stress in the heart. We didn't see that effect in electronic cigarette without nicotine. Therefore, nicotine is relevant for this effect. When the mice were exposed for 12 weeks with the saline electronic cigarette without nicotine, electronic cigarette with nicotine, we observed a cardiac dysfunction that without changes in the morphological parameter of the heart. Here's the echo data that from that experiment, where we see that the decrease of fractional shortening, decrease of velocity of circumferential fiber shortening in mice exposed to electronic cigarette with nicotine. As well, we observe a decrease of ejection fraction. Well, adipocyte tissue not only release free fatty acid, as well release adipocyte tissue release another molecules that are bioactive. For instance, adipocyte tissue, when the adipocyte tissue is dysfunctional, it have adipocyte tissue remodeling, and it's characterized for inflammation and increase of infiltration and polarization and average polarization of macrophage that can lead to metabolic associated cardiovascular risk. As known, the free fatty acid release can activate the toll-like receptor 4, TLR4, and lead to the pro-inflammatory response, leading to the accumulation of the macrophage in adipocyte tissue. It's known that Citrun-1 serves as a protector against metabolic disorder in the multiple tissues, including adipocyte tissue. In adipocyte tissue, Citrun-1 control macrophage number, and Citrun-1 adipocyte tissue specific knockout is susceptible to diet-induced insulin resistance, which is associated with the increased number of adipocyte resident macrophage. Here in this Western, we show that this is a Western blot of the mouse exposed to electronic cigarette without nicotine, and saline. And we see in the Western blot of the adipocyte tissue that the electronic cigarette induces a decrease of the Citrun-1 in the adipocyte tissue. In the right side is the quantification of the data of this Western blot. One of the characteristics of the adipocyte tissue remodeling is, as I said before, is infiltration of the macrophage. So we stain for a marker of a macrophage, F480, and we see infiltration of macrophage in the adipocyte tissue. Additionally, we stain for mesothelioma stain in the adipocyte tissue and pseudo red staining, and we see increase of the marker for fibrosis. Then we take a piece of the adipocyte tissue and we perform a transcriptional analysis. And here we can see in A that the two-dimensional hierarchical clustering that compares saline versus electronic cigarette with the nicotine. And we see there is a change of 572 genes. When in B, we compare from the 25,000 genes that we sequenced, we see only 93 genes that change when we compare saline, again, electronic cigarette without nicotine. And in adipocyte tissue, when we compare electronic cigarette without nicotine versus electronic cigarette without nicotine, with nicotine, in C, without nicotine, and compared with the electronic cigarette with nicotine, we see a change in 561 genes. Well, now we want to determine which genes are nicotine dependent or nicotine dependent. To determine which genes are nicotine independent in D, we compare saline versus electronic cigarette, 0% versus saline versus electronic cigarette with nicotine. The change in the same way are nicotine independent. We observe only 28 genes that change in that way. When we want to find how many of all these genes from the original 572 are nicotine dependent, we compare, and this we can see here in E, in this Venn diagram, that we compare electronic cigarette with 0% nicotine versus with the data of electronic cigarette with no nicotine versus the one with nicotine, and we compare with the one who could change in the same way versus the saline versus electronic cigarette with nicotine. From the 572, we observed that 294 change in the same way. Therefore, a major number of the gene change in adipocyte tissue are related with the nicotine change. That can be corroborated by the effect of, we can see in F, the PC analysis. The PC analysis basically is a way to reduce the dimension of the data and to see if a group is different to the other we can make a circle around the data. And you can see here in yellow that the mice exposed to electronic cigarette with nicotine are different than the others two groups. So you can see a circle, we can circle all of them. We have started to, well, we haven't finished this data, but we have started to validate some of the genes that change and they are nicotine dependent. And for instance, we have seen increase of CD4 that is in mouse is a T cell marker, and as well we have seen increase of endothelin that is substance that is released by the molecule that is released from the adipocyte tissue and is involved in vascular vasoconstriction. So when we put all this data in HIP analysis, that's a web-based analysis, we see that one of the major characteristics that they show this analysis is we see an increase in the addition of a pathway associated with addition of agranulocyte and granulocyte. So therefore, there is increase of this inflammatory pathway and increase of the inflammatory cells that go inside of the adipocyte tissue. Let's go together with the dysfunctional adipocyte tissue. So as we explained before, free fatty acid, we believe that free fatty acids are relevant for the function of the heart. So we use a drug that called acipimox. Acipimox is a nicotinic acid derivative that inhibits lipolysis in the adipocyte tissue and hence decrease the free fatty acid levels in the blood. And as well, we know in our lab have published before that the acipimox reduced nicotine plus high fat diet induced hepatic steatosis, a common antecedent of metabolic syndrome. So here is the data from the effect of acipimox and the electronic cigarette induced cardiac dysfunction. And here we can see that the mice were exposed for 12 weeks to saline or to electronic cigarette with nicotine or electronic cigarette with nicotine plus acipimox. And we see that the 12 week as before, we induce a cardiac dysfunction phenotype with decrease of fractional shortening and a ejection fraction of velocity of circumferential fiber shortening. And we see that the acipimox inhibit the totally rescue the cardiac dysfunction phenotype induced for electronic cigarette. Well, adipocyte tissue not only release free fatty acid, as well release cytokines, and some of them are related, are being implicated in inflammation and cardiovascular disease. For instance, interleukin-6 mediate inflammation leading to some of the cardiovascular complication in metabolic disease. As well, adipocyte and macrophage residing in adipocyte tissue are the major source of the elevated plasma concentration of interleukin-6. And interleukin-6 have been identified as an independent predictor of type 2 diabetes and associated cardiovascular event. As well, macrophage colony stimulatory factor, MCSF, is a multifunctional pro-inflammatory cytokine that regulates differentiation, proliferation, and survival of the monocytic progenitor cells. And the MCSF have been shown to play a role in the in the formation of arteriosclerotic lesions. So here is the data from the mice for the serum of the mice that were exposed to saline, electronic cigarette, or electronic cigarette plus acipimox. And we see that the NA, that the acipimox decreased the level of the free fatty acid in the blood as expected. And when in B and C, we see two analysis of MCSF and interleukin-6 that come out from a panel or array of cytokines. And we see that in B, that the mice exposed to electronic cigarette have increase of MCSF, and ACP-MOX decrease the levels or rescue the level to the basal. As well, very important that the electronic cigarette induce that the increase of interleukin-6 in the blood, and ACP-MOX rescue or decrease the level of interleukin-6 in the serum. With all this data, we can produce a working model where electronic cigarette activate lipolysis in the adipocyte tissue. The adipocyte tissue is known that after activation of lipolysis, recruited macrophage and lymphocytes. And now the adipocyte tissue release adipokines and of course release free fatty acid. And this lead to cardiac dysfunction and an inflammatory metabolic phenotype in the heart. So at the moment only we have tested with ACP-MOX the relevance of lipolysis for this model. The conclusion of this work is basically electronic cigarette with nicotine induce a decrease of CYP1 in the adipocyte tissue associated with increased free fatty acid marker of an increase on the marker of adipocyte dysfunction. As well, a transcriptomic analysis showed the deregulation of inflammatory and increased changes in the granulocyte and granulocyte adhesion pathway in adipocyte tissue in mice exposed to electronic cigarette. And we had shown as well that ACP-MOX prevent cardiac dysfunction induced by electronic cigarette in mice. ACP-MOX will reduce the circulation level of free fatty acid and cytokines induced by electronic cigarette. Here is we want to thank my advisor, Dr. Friedman and Dr. Siha Hikim, and Dr. Shao that worked with us, of course, and Dr. Jordan and Dr. Ross that have worked with us in to do the echocardiography, and in the RIAR, all the students that work in this project. Thank you.
Thank you very much. It was a beautiful talk. We have questions, anybody?
Jorge, this is Rajan. I have a question. Did you look at the effect of ACP-MOX on the adipokines in general?
Well, the array that we ran some of the adipokine, but not all of them.
Okay, because
that's an array.
Because you're talking about this SIR2-in-1, so most of this SIR2-in-1 and free fatty acid, it goes back to the adiponectin level. So I think it will be very important to connect that link if possible. So that was my question one. And another question was, you have looked at the effect of ACP-MOX on e-cigarette. Did you looked at the effect of ACP-MOX in combination with nicotine? So e-cig plus nicotine, in that group, did you look at the effect of ACP-MOX on any of those parameters?
Well, the ACP-MOX is together with the electronic cigarette with nicotine. If your question is,
okay. The way you labeled it looked like e-cigarette. So this is a combination, right?
Yeah. Basically, it's the mice were exposed to electronic cigarette with nicotine plus ACP-MOX. And the lab before had done injection of nicotine intraperitoneal, and as well have seen an effect with ACP-MOX.
This is identical effect.
and not in the
Identical
heart.
effect,
Yeah,
both on
basically.
skeletal muscle as well as liver. Yeah,
Yeah,
you're right.
in the liver, but they didn't check the heart and the.
Jorge, I have a question. This is very interesting data. These are ApoE mice or wild type mice?
Okay, the original data is
No,
ApoE mice.
these data you showed about acipimox.
No, the acipimox data is wild type and the wild mice.
So, you know, the reason I'm asking this question, I saw you have very marked increase of MCSF1. And that part, one of our faculty, Drew faculty, he used to be also in CEDARS, he did a lot of work on MCF, and MCSF1 is directly related to the atherosclerosis. So I don't know why, because these mice right now, we didn't see that much of atherosclerosis. But what this MCSF1 is doing, what is your conjecture regarding this?
Wild-type mice are resistant to atherosclerosis, you know. So, but for the way, it doesn't mean
increased MCSF1.
Yeah,
how
but
is
doesn't mean they might have a role in the mice who can develop atherosclerosis. You know, but MCS1 is kind of difficult because of very pleiotropic effects. So for me, the one who have been this comes from an array, so I didn't pinpoint this to follow it. So, but interleukin-6 have been a lot of work. It's much more work in that about the effect of interleukin-1 in the effect in the cardiomyopathy and especially in metabolic disease.
Right.
So
we have seen that in muscle as well as in the liver.
But so, I think the effect of IL-6 in muscle versus other inflammatory diseases are quite controversial.
Yeah, in general, all the cytokines have multiple effects. So, but for whether in humans, high level of interleukin-6 are associated with higher risk of cardiovascular disease. So, so there, it's hard to, you know, the cytokines, but the cell has a very pleiotropic.
Thank you, Jorge. Good.
Jorge, just 1 second. You know, since you are talking about these inflammatory signals, and since you have rightly identified IL-6, so I'm wondering if you could just extend this thing to a little bit like IL-6 works through this IL-6-1 receptor alpha, and then it fits into this STAT6 signaling and into your toll-like receptor, which kind of fits back to your inflammatory like signals. So probably it may be useful if you if all these things are available to you, you can correct.
Well, you know that we can use maybe, I'm not sure if we can in the future try to use inhibitors for the receptor. You know, that will be a possibility. So what I want doing now, actually, Candice here, she's trying to do is to see if we have a marker for inflammation and the increase in the heart tissue. So still, you know, this data is not published. We have to see, and especially they're basically more the being critic. Big part of the data is associative mainly. So the only data that we can claim cause effect is about through lipolysis, you know.
Yeah.
So it might be a change in the cytokines associated with the disease, with the changes in the function of the heart, but doesn't mean necessarily a cause effect.
Yeah, most of
I will expect. I hope I will try that to do it, but still we are not sure of that. So that will be tested. We can test in the future. That's maybe.
Do you want to comment on clinical relevance of your talk?
Well, free fat. Okay, there's a dreaming a little bit in the long term. We can set maybe treat the effect of the electronic cigarette through acipimox. However, there's some work in the acipimox with patients, and when they use acipimox with patient, at least the paper has seen it, they improve the lipid profile, they improve the level of glucose, but however, at least in patient they didn't change with the cardiomyopathy, with the idiotopic cardiomyopathy, they didn't change much the contraction of the heart. However, might be possible if there is we are dealing with two different mechanisms of them, but it might be, it's a possibility that it can be analyzed, but it's a long shot, of course, from that.
So to add, Acetamox was a medicine used to lower cholesterol.
Yep.
Right.
No longer used in the United States. It might be used in a few places in Europe. I tried to, first of all, it was made by a company called Pharmacia that's no longer in business, and it's been bought out by Pfizer. I originally tried to have Pfizer give me the rights to use it in patients, and that didn't work. And I went to the FDA to try to get FDA
Your
approval
voice is
for
breaking
it,
up. Theodore,
and that
voice
didn't
is
work
breaking
either.
up.
So yeah, sorry. So I tried to get FDA approval to use Acetamox in patients that are smokers, and I couldn't get FDA approval for it.
Well, maybe you can try with electronic cigarette.
Well, no, the issue was the Acetamox. They wanted all the safety data about it and all this data that I didn't have, and the drug company basically didn't respond to me. Any other final questions for any of our panelists today? Okay, if not, we'll close. Again, I thank the panelists for talking. Everybody, was very great symposium. Thanks all our SART trainees for attending. I hope you learned something. And again, April 16th is our next SART Institute, and May 7th is our next e-cigarette institute. E-cigarette.
Very good.
Okay,
Very good.
everybody.
Okay.
Yeah. We'll see everybody later. Thanks.
Okay.
Thank you. Thank you very much.
Bye-bye.
Thank you.